Can you remind me that I need a photo? A photo of you? Oh, Andreas, I also don't know your family name?
Simon, S-I-M-O-N.
Okay, guys. Hello. Welcome. Close it, don't worry. So today we want to talk about the effect of sequence diversity in people. Again, let me just repeat, there will be no lecture next Thursday. There will be a lecture on Tuesday, but not next Thursday. Special lectures currently still unclear. This date is now confirmed, 28, 11:30. We still look for a room that will accommodate enough people. Other than that, this is what it is. Thanks to Christian and Dimitri for the video lectures. For us there was no last video lecture. They promised that this will be available soon. The slides for today should already be online. Okay, personalized medicine. What do you associate with that term? Anybody? Nobody associates anything with that term? Guys, what's happening? Really? Is it boring or?
That you treat a person medicamentously based on its genome and individual based on its genome.
Yes, and that in fact is exactly the way it was coined, and that is the way it also offended many physicians who felt, what do you mean? We have been doing personalized medicine since many, many years. We look at the patient and we treat them individually. But ultimately that is in fact exactly the idea that is behind it, that you somehow, from gazing at the genotype, you learn something about the phenotype, you learn in fact to design a drug that is specialized to an individual, to the genotype. And in order to do that, of course, you need to address many levels. You need to sort of look at the molecular level here, RNA, proteins, signaling networks, cells, organisms, and the environment. And essentially, the effect of a drug plays on all of these levels. Okay? The lecture, protein prediction, will sort of play on this level. We will talk about effect on cells, we will talk about somehow signaling networks, about proteins, about how we can predict from sequence, in some sense that comes from here, how proteins act. Sometimes I will go into the effect upon an organism. In fact, today is one of those examples where I will mention that, but I will not talk about microbiomes and the like. Okay, now the human genome is completely sequenced. Since when?
2001.
That's a good guess. Very good guess. So it was supposed to be sort of the event for the new millennium, was supposed to come out on January 1st in 2000. Came a little bit later. Here's the announcement of the event in June, where we see Francis Collins. Anybody knows who? So there are three people here. Bill Clinton, I assume that everybody in the room will know this, the then US president. This is Francis Collins, and this is Francis Collins, and this is Craig Venter. Anybody knows anything about Craig Venter and Francis Collins? Or either of them? So Craig Venter started very early on to invest into sequencing genomes of entire organisms. The first entirely sequenced organism, Haemophilus influenza, was done by his company. He started a company to do this because he could not get an NIH grant for that sequencing. Because of a variety of reasons, and he simply decided, I'm going to turn to private investors, and I'm going to do this by private donors, private money. And he has been doing that ever since. And it was his company, Celera, that sequenced, or was one of the sequences, the first human genome sequences, was from his company. The other side to the first genome came from all the public efforts, and the public efforts were coordinated by Francis Collins, who now currently since 2 or 3 years, I can't remember, or longer even, heads the NIH, the National Institutes of Health in the US. Do we? Let's just look at the numbers. The DNA library is sort of 3 billion letters. It's a 3G, and this 3G fills exactly those 4,000 books. So I counted for every single one of those the letters, and so this is really true. Essentially, I did not count every single letter, okay? But other than that, I'm very confident that what I show you here, this library, contains exactly sort of 3 billion letters. And I made a I spent some time on this. On the other end of this story, we have the machinery of life, the human proteome that we currently believe is about 20,000 proteins. We do know there are about 100,000 constructs that we can experimentally determine, that we can experimentally measure. But whether the difference between those are alternative splicing events that are functionally important, we still don't know. This is sort of a relatively recent number here. Shortly before the human genome was sequenced in 1999, people believed we would have 100,000 proteins. So we have the 100,000 magically back in some sense. But in between, the number of proteins in human has shrunk. So when the human genome was published, it was believed it was 30,000, now it's 20. And if anything, it looks like the number is going to go down a little bit possibly. So this is constructs. Alternative splicing variants is one explanation for that. Okay, there are others. But now one big issue when the first human genome was sequenced was this assumption that one human genome is enough as a reference genome. And when we look at a population, we're going to just look at differences to this reference genome. Okay? We don't need to do another human new assembly. You guys understand how you assemble shotgun sequencing? So shotgun sequencing essentially cuts short fragments of DNA, and a novel assembly of a genome simply is that you have to find a way to put these pieces together. How can you hope to do that? Well, if you have a reference genome, you can fit them into the reference genome, right? But novel assembly means you don't have a reference genome. How can you then do it? Is it too trivial or is it too complicated?
They do it with the Queen class, so they have class and search for overlap between the
Exactly. You have, it would not be, if all the fragments, if the fragments that you have wouldn't overlap, you couldn't do it. The only way you can do it is you have overlap, and you create overlap by simply having random cuts of a lot of fragments. Do you have any idea? So ultimately, so sometimes you may have situations like this, then we have situations like this, we have situations like this. So this particular case, essentially for this particular region here, we would have 4 sequences, or 4 fragments that overlap. Do you have an idea, in order to get a good human genome today for one person, how many times you have to have overlap? What is considered good?
100.
So 100 is considered very good, yes. In fact, the number 100 is only on the table because sequencing has become so cheap that we can afford the answer 100. But just a few years ago, what is called 20x was something that was a major publication if you reach that. So the first sequences they had, they just had numbers less than 10. Now, who do you believe was sequenced in this event that I showed you in 2000 that was celebrated? Do you believe this is true? Why? Well, good. Okay, there's a company. A company can do that. Yes, that was one answer. What about the public? Francis Collins? If you ever read a book from Francis Collins, you would very, very quickly realize he would never do that. But then, public things don't work like that. So how do you believe they did it? There was a committee that chose a representative set of people, and from everybody came something. And the people were not documented, made publicly available, so nobody really knows. It could have been one. Okay, we don't know. But there is strong evidence that there is a variety. So some people sort of dug down into the data to establish something about how much you can see in a genome. And there's clear evidence that this is several individuals. But all of that got us going into this idea that diversity sort of may play a role, and that maybe even if there is one reference genome and everything is differential to that, it still would be interesting to see what the differential to that is. That got the HapMap project and the 1kg project off the ground, launched in 2008. As the number says, the idea was to sequence a diversity of people, 1,000 people. Okay? Then other projects began in England, 1K UK, 1 million Chinese. There are two new initiatives. One is in April 15, Genomics England, and that is 100K plus medical data. And in January, historically before that, not April 15, around about mid of April. I'm not sure whether April 15 is right. January 22nd, 2015 is what I meant here. But January 22nd, 2015, Obama had in his address to the nation the precision medicine address. By the way, it's very interesting to listen to that. You can easily find that State of the Union address from Obama this year on precision medicine. The dream to essentially pull all the resources on computers and do a better medicine through genetics and combining that with a lot of additional data. Here we talk about 1 million. The difference between the precision medicine that is in January and the Genomics England that is in April is Genomics England has the money. In the middle of April, they got the money. Anybody any idea what we're talking here? The money? Dimension, ballpark, 0.5 billion euro. That's the amount of money they got on their bank account in the middle of April. So here, the amount of money is bigger, but at this point is not yet on any bank account, while this one is already operating at very, very full speed. So they really have funding, and they will realize that within less than 5 years, both of these initiatives. Both of these have then to do with diversity. And that brings us to the question, how much do we differ? So the entire course that we will have will be on proteins. So my question about difference will be a question on the set of coding. So for how many of the coding part, so how many of the proteins will we have an amino acid change? What do you believe? Let's just, you look at your neighbor, either way, either left or right. What will be the number?
10,000. Synonymous changes.
So amino acid changing, that's exactly what I'm asking. 10K. So again, that is what, sorry, it will take a while until next year when you believe that I remember your names. Michael, so Michael claims to have 10,000 variants with respect to everybody in the room. Okay? Anybody feels this is high, feels this is low? Who jumps and says more? Who jumps and says it's less? 2. Everybody else doesn't jump. Because we should open the window, or I'm not motivating enough. So clearly, I don't know what you guys know, clearly at the point at which all of this was started, the assumption was much, 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 much less than that. If in 2000 somebody would have said, I expect 1,000, I believe everybody would have jumped up and said, no, 1,000 SNPs maybe. Or maybe 10,000 SNPs, but not 10,000 amino acid changing ones. That actually was considered a very high number. But the fact is, the number is higher. It's close to that, it's the right ballpark, but we differ essentially in one amino acid in every single protein that we have. Okay? Here, I put it into the Bavarian coat of arms, the difference. The ratio is such that the blues are where we are alike, and the red is where we differ. Okay? Now, this, I'm not entirely sure, will strike you as a large number in some sense. It's mostly not really, there's not a lot of red here. It's mostly blue and white. White doesn't count. But don't forget, that's behind this library. Not quite true, because this library is really 3 billion letters. And what we are talking about is the coding part here, which is about only 1.5% of that, but still. And the other thing that you have to also realize, somebody with my body has about 55 trillion cells. And essentially, I have the same genome in every one of those. Not one difference. Okay, there are some differences. But in principle, the answer is, for my 55 trillion cells, there's not one change. To any one of you, there's 20,000. So this is a big number. And this big number started to make people realize that there's much more diversity than we had anticipated. And the diversity comes on different levels. So one level of diversity is epigenetics. Epigenetics is essentially the issue about changes in your built-in organism or structure that are inheritable. Changes on the histone structure. Changes that you will pass on to your offspring beyond your genome. That will change while you live. Yes, changes in our genome will come by radiation. They will come from extreme chemical stress, and those kinds of things can make changes in my genome. But most of my daily work, of my daily being, mostly I will have the same genome when I'm born and when I die. The telomeres will shorten, maybe there will be some mistakes, but essentially not much will happen. These things will be affected, okay, on a per day. Now, the diversity in some sense has led to a situation in which we can study migration of people. We can study populations. Here is a paper, and by now it's a little bit, it's not anymore the recent, it's 6 years ago, but I found it amazing. You can make a statement such as this, you find a little bit of biological evidence somewhere in the ice, and you can make a point that there was a migration from Siberia to the Americas before 5,500 years. 5,500 years is when we believe that the current, or the native Indians, the Native Americans, those were originated from a move in 5,500 years ago. And there was a move before that. And you can find out from genetic evidence frozen somewhere in the ice. It's amazing, I find. And ultimately it comes from diversity. Here's another step up in complexity of, or in awness, for me at least, Svante Pääbo, who is running the Max Planck Institute in Leipzig, and I keep forgetting Anatoly Derevyanko, in a cave in the Altai Mountains in Russia. That he's the one who found body part that the lab of Svante Pääbo sequenced. It found out this is a new hominid. They termed Denisovan, because this is, the cave is in Denisova, the Denisovan cave. And it's the first time in history that a species has been coined according to genetic evidence. All the genetic evidence of the Denisovans has disappeared. Essentially, it was ground up to do the sequencing. When this sequencing was done, the Denisovan, at that point, was the best sequenced creature in the world. So that was in fact one of the first 30x sequencing that had ever been done. And for people that nobody had put that much effort into it, simply because that was all they had of it, so they did it again and again and again. But in doing it again and again and again, they ground up the material. That in fact had not been classified as human, had been classified as cave bear. And I can't remember how they stumbled over the cave bear, why they ever sequenced it. Maybe I can't remember the answer to that. Anyway, so that is clearly an amazing part. Now that brings us to this issue that in principle we believe that from the differences between us are imprinted upon the differences in the genome. We in principle believe that, let's begin the story here. So we have two soccer teams here, the Swedish, the Italian. You assume that there are genetic differences between the Swedes, and you assume that there are genetic differences between the Italian, but you assume that essentially they agree more with each other than any of the players between these two teams. And you would assume there's even more within one team, and even between these two teams with respect to each other, so the Italians and the Swedes are more similar to each other than any of them is to the Chinese. And then you would sort of do the same, you would assume the same thing for instance, here the Africans. What I show here are 4 Bushmen from the Kalahari in South Africa, very close to where we believe Homo sapiens started. And they all have, for me, a very similar physiognomy. I would not be able to distinguish them. They have a similar size, they have a different, similar life conditions. And again, I would very clearly say that if I sequence those 4, yes, they differ. But if I looked at them compared to those groups, then they differ less from each other than we differ from them. Okay? That's what I would clearly say. And that is entirely wrong. So the sequencing was done, Desmond Tutu was another one who was sequenced here. There was a remarkable story with this, but I'm not going to talk about that, from Vanessa Hayes, or the consortium led by Vanessa Hayes. And they found out that in fact the differences between those 4 were way, way, way higher than the differences between anybody in this room and the Chinese, or anybody in this room and the North American or Western European. That's remarkable. Now, as science always goes, the moment you have an observation, you explain why this is the case, why you had always anticipated that. In this particular case, the way this is explained, I don't consider this an explanation. This is what I wanted to make clear. But what currently everybody believes is the answer to this conundrum that we didn't expect is when you have a group and you have a certain diversity in this group. Now you have an out group, so some fragment, some subset here that leaves Africa. Okay, it's a little bit the wrong direction. So they go here, they go to Mesopotamia. And they go, well, pretty much it's not that wrong. They most likely go from the eastern part of East North Africa to Mesopotamia, so that somehow is the arrow, that somehow is the direction. This outgroup has a lower diversity genetically than the entire group. Let's just think about, for instance, rare variants. Variants that are carried by very, very few people. Say, initially in the entire population have 100,000. Let's just assume there are some variants that are only in a particular person. Say this is 95,000 here, and this is 5,000. The outgroup 5,000 go, 95,000 stay. And by that definition, you already have at least 95,000 variants. Say each had one variant that is only for them. 95,000 do not go out. Okay? So you already, the group that goes out has a lower diversity. And this lower diversity will essentially not increase unless you have a very short generation time. So in yeast and fly, it changes. But in human, where we talk about this event happened, does anybody have an idea when roughly this happened? What the current estimates are for when we left? So again, there are 2 different models. There is an alternative model that Homo sapiens evolved simultaneously at different places on the globe. This is many people in China believe in this model. The ruling Western model is really that there was an out of Africa movement, and in this view of the rule of out of Africa movement, clearly that happened. Maybe there were people already in China then. But even if that happened, do you have any idea when roughly that did happen? Again, it's also now clear that people came back to Africa.
20,000 years?
You said 20?
30?
Yes, that is not that far off. So people, and in fact these numbers are in flux. I'd rather say that we would possibly want to make this somewhere between 30 and 70. And one reason is, again, going back to not this group here, to Sante Pabo's group, recent evidence. You know that everybody in this room carries Neanderthal genes. Do you know that?
For what?
Neanderthal?
Oh.
Everybody knows that, right? Do you have a problem with that? Remember the definition of a species in biology? What's the definition of a species? A speciation event. So you have a P and Q, and they get child. This is the common event, and then children mix. But if we have an offspring of a species, then in fact we create sort of PC and QC. What's the definition of a species in biology? It is that when you have an offspring between PC and QC can have offspring. They can mate, but the offspring doesn't survive. It's infertile, right? And horses and ants, donkeys is one example. Mules come out of it. Mules don't have children. So biology is full of these examples. But the fact that we carry Neanderthal genes implies this is not always true, right? Obviously, there are offspring of cross-species mating, which in fact, again, is not only that it may not make us feel comfortable, it is a violation of the species concept, okay? So it's a major finding to make that point. Now, the question is, when is this mixture, when did the mixture happen? So the offspring between people and Homo sapiens and Homo neanderthalensis. And there is a paper from the Pabo, from Svante Pabo and Janet Kelso, that says the point is pretty close to 50K. So the mixture was about 50,000 years ago. And that puts a clear, if you believe that point, that puts a clear stop into the original idea that maybe it happened 30,000 years ago. So your number 30 has been quoted, has been believed for a while, but it must, maybe briefly before that, maybe it is 50, 51 or something. Maybe it was, they directly ran to the north to the Neanderthalensis, mated, and then that happened. But actually not quite, because that happened somewhere in Russia. This event happened somewhere in Russia. Anyway, so all of this we can trace, and I believe there will be much, much more, and we already see that over the last years, is a study of differences in the diversity. Most of this study, however, is one that has to do with the non-coding parts. And the non-coding parts have a different evolutionary constraint, and that's why the movement of species or the movement of populations are mostly traced by non-coding variants. Now let's talk again about the coding ones. We have 20K between any pair in the room. And the question is, do they have an effect? Okay, let's look at extreme effect. How many of these 20,000 can be absolutely lethal? Can anybody give me an estimate on that? So definition of absolutely lethal, you die. Carrier dies. How many of these can be like that? I did not provide you with the information to answer that question, but implicitly I did. Think about how sequencing is done for the 1,000 people. How do you believe 1,000 people are sequenced? What's going on? Are we all together in the room, or so you walk in? How do you imagine that you are sequenced?
They give away something of their DNA, which means they are essentially alive.
That's the important part. In fact, now there's sequencing done on corpses. People are dug out for a variety of studies. You may have seen the Black Death that recently was published. There's a lot now done that has not been done before. But what I'm talking about is all people who walked into a room and did their sequencing. So we already know there's no killing lethal mutation in there. Okay? That's an important thing to remember. Which means it's not a set of all the kinds of variants that you may observe in people who are even born. And clearly not all the kinds of variants that you may have. Okay, so we could address that experimentally. For how many of these do you believe we have an experimental answer? So it's just the original data set that I'm going to show you plots for, all based on the original 1,000 people sequencing. It's a subset of what is available today. It's just that we have a long project here, and some of the exercises will focus on the same data set. And in that data set, we have roughly 300,000 variants. Okay, 20,000 per person times 1,000 is a little bit more than that, but this is simply explained by the fact that you, in many cases, observe the same variant in more than one, or you share variants. So there's about 40,000 variants that are common. Common defined as more than 5% of the population. So there we already have the count back almost entirely. But anyway, so this is what we're talking about. How many do you believe of those are experimentally covered? 400,000. Of 400,000, how many do you have experimental data?
1,000.
No, way less. So 40. 40 is a push of the story. At the moment. Again, at this point, people now begin to sort of systematically target some of these proteins that are involved and do deep mutation studies. So that is beginning to change. That is a change that has just happened in the technology over the last 3 years, so it's fairly new. But ultimately, if we wanted to answer that question experimentally, maybe we have to wait until 2050 or something. That's a provocative statement. What we can do today to answer the question whether they have an effect is we can use computers. And that again is the method that we briefly, the type of methods that we briefly discussed about on Tuesday. By the way, I had to give you another thing. ACE-DB is a database of molecular alanine scanning E database. The E, I forgot what the E stands for. That's simply where you have a reaction between two proteins and you measure the delta G of binding of an alanine, mutating that variant to alanine, that original native sequence to alanine. I just remember that I owed you this name. So we can use a computer. We have a method. Here's one, and we get to much more detail about that has been developed by Jana Bromberg, who is at Rutgers University, and also an IAS fellow, the Institute of Advanced Studies at the TUM. Here's the logo of that one. Together with Max Hecht here in my group. Essentially, it's a neural network that uses the variant and some information about the variant as input, and then predicts effect or not effect. Effect may mean that this is the original. In some sense, that's a visualization. It predicts the effect on function, not on structure. So if that were the original you came in, effect can mean that you sort of break it apart, or it could mean that you just do it differently. Actually, gain of function. It's also an effect. Loss, gain. Both of those would be predicted as an effect. Method cannot distinguish. And then neutral, the variant doesn't do anything to it. Now, this is a cumulative curve of the SNAP output score. The SNAP output score is scaled to 100 minus 100. 100 means very strong prediction of effect. Minus 100 means very strong prediction of neutral. I show you cumulative curves that answer the question, what fraction above a certain threshold, what is the effect predicted for that fraction of variance? For instance, if I had a data set experimentally known to have an effect, if SNAP were completely fantastic method, always right, then it would predict essentially all of those variants here, 100% of those variants almost, with a very high score. Just for instance, 98% of the variants would be predicted at a score of 82 or higher. Okay? Then it would be a good method. If I started with a set of neutrals, and if it would be a very good method, then would, for instance, say that only 1/4 of all the variants are predicted at values higher than minus 90. So 1/4 is on that side of the distribution, meaning that 3/4 are on that side of the distribution, meaning that 75% of the variants are predicted to be less or more neutral than this. Or looking at the zero sort of threshold, almost everything here is predicted left of that zero line. Almost everything is predicted to be neutral. Now, this is computational biology, this is biology, this is not as good as SNAP is. This is the reality, that's the development center of SNAP, and that's an experimental effect for cross-validation, that's experimental effect for neutral, experimental neutral in the cross-validation data set. So the differences between the sort of ideal and that would be here or here, this is the mistake of the method, and conversely, the degree to which red and green are distinguished is the power of the method. If you had a random distinction, the random distinction would just fall here. Not a random variant, but a method that predicts random. They would just not make a difference between red and blue, red and green, and therefore sit somewhere in between. Now, what the method learns, and again, I will have several lectures on explaining this method, what the method learns is the prediction of a molecular effect, in principle. The ASDB kind of thing. You measure the effect of a variant on the binding of another protein or a substrate. When you now look at variants that cause disease, I can't remember, somebody, Michael, were you the one with OMIM? Somebody threw OMIM, or Hauke, were you the one? Somebody threw on Tuesday threw up, when I asked what database could we use to develop a method that predicts the effect, somebody shouted OMIM. It was you.
I guess he's not here today.
Oh. Niklas? Oh. Was Niklas?
Maybe it was me, I'm not sure.
You have to stand up to your word, come on. You know, once in a lifetime I remember what had happened yesterday, and then this is so not fair to your teacher. Anyway, so we have a lot of variants that are of the type a single variance causes an effect. One single sequence variant is going to be having a macroscopic effect on the organism. Now, let's just pause here. We predict molecular effect. I predict this variant in that particular protein, is that going to change the function of that protein? That may or may not translate to an effect upon the system, right? There may be all kinds of buffers. There may be another protein that I have to do this. So if I now started with an OMIM data set that has not been used for training here, an OMIM data set that is such that I have 6,000 variants that cause a phenotype, very often a disease. Sometimes it's not quite disease, and we will get back to that. What do you expect will happen? Where will the curve be? So far, I show you data of 40,000 variants that had an effect experimentally that were used for training, 40,000 roughly neutral, also used for development. And this is a cross-validation. Now I'm bringing a new data set. I actually don't know anything about the molecular effect. All I know is they cause an effect on the organism, disease. Where do you believe this will be predicted? Anybody? Let me just...
No mistakes because you don't know the set and the set also.
I don't know, the set is true. I'm sorry, I have a problem to translate your movement with your hand into... how did you move your hand on this?
I mean more in the...
Here? Here. Somewhere here. Let's not discuss. But you say it will be a little bit below, it will have a shape somehow more like the red than like a green, but below the red. Is that a fair way of interpreting your hand?
Yeah.
Does anybody doubt this expectation? This is absolutely what I would expect. This is what Chris Schäfer has observed. The orange is omim, is above. And for us that was very remarkable, and again part of your exercises will have to do with sort of looking at some of these things in more detail. It was remarkable because it doesn't happen very often when you develop a machine learning tool that you come up with a new data set, and the new data set somehow, if you believe that clearly they should have an effect, clearly this is the kind of thing you dream to capture, that this effect is even better than your development set. You do better on a new set than on one that you developed. That's unusual. Our interpretation for that was that the orange is just more consistent. When you measure the effect upon a change of a protein, you may make mistakes. Disease is just such a big effect. There's nothing about mistake here. It's a very, very strong proxy. So the consistency of the orange data set is much higher. Yes, you have sorry, you have cases where you have alternative function, but if those cases are not there, otherwise there would not be the disease. So we have very clean data, and that's obviously captured by the system. Now that it means, in turn, we now can address this question and simply ask how these 20,000 differences matter. And by the way, I show these three guys because it's a longer story, and we have been sitting on this manuscript for years. It started with Dominic, with a master's thesis of Dominic, went into a master's thesis of Yannick, who now does his PhD on somehow at Rutgers on related issues, and Max is still working on it, and we hopefully send the manuscript this week. Anyway, it's a long, long story. So the question here is, now, if I looked at these 20,000 variants, where do you believe they fall?
Questions where the curve is for 20K, which differs between us. According to the effect of OMIM, I would expect it on the left side of the green curve, because it's a quite clear data set of
so where
enough to lead for mutation.
Thank you. Anybody else? Any opposing view on the matter? Again, in both cases, I can tell you. Madalina. So I completely share Madalina's view. Okay, you say it?
Madalina. But it's Mada, it's perfect.
There's always this story sometimes. Yeah, Madalina's answer for the previous one is what I would have expected for the orange, and what you said I would have absolutely expected. I believe, being a machine learner, I would have never put it below the development set. I would not. In fact, then initially when we saw the first time the graph, we had not seen this one here. So I believed it was slightly above the green, but essentially where the error points. That's where I clearly expected it. Okay? That's where we are. Now that, unfortunately, is why this took so many students. Because the supervisor never believed the students. They came up with a master thesis, and then they had to do it again, and they had to do it again, and I still didn't believe it. It's not true that I was the only bad one here. Whenever I ran around and presented the data publicly, people didn't believe me. So I went back, hey, we have to redo that. And this is why it is dragged on for such a long time. And we have now a bunch of findings that are really punches, that are really, really amazing, and this is just one part of that story. So it's just somehow in between these two extremes. And that clearly is amazing. Now, it means, for instance, that for 1/4 of all the variants, we have something that we would consider a score of 40 and higher. We would clearly consider a very, very, very strong score. Okay, let's sort of step out a little bit and try some other data set. Those are the variants between people. What would happen if I looked at the variance between us and, say, Denisovan or Neanderthal? Actually, what I'm going to show you is a mixture to both of those because of technique. No, it's Denisovan what I'm going to show you. What do you expect there? So let me explain to you how I have to do this. Now, I take all the proteins between human and Denisovan, I line them, and I'm going to look for frozen differences. Frozen differences are defined as, in this particular case, the way we did it is we, in fact, not we, that comes from Janet Kelso in Leipzig, in the Max Planck Institute in Leipzig, and so the way they look at the data, they always say, okay, what we care for, and that went back to the Neanderthal, first Neanderthal genome, what we care most about is how Neanderthal and we differ. In fact, what we care about are things where, say, Neanderthal is like chimp and different from any human we have observed. Those are variants for the first Neanderthal sequence. Those were actually a bunch of less than 100. It's a very limited set. But those kinds of things we care about looking at to understand differences. And we haven't made a big step yet. But here we are now back to, we look at all the proteins where Denisovan and human have a protein that is recognizably identical. Then we look at all the residues that differ, and we call them variants. Okay? Where do you expect it?
I would expect it to be random.
That is, you push me to a point, that's a very, very good point. And the problem, Jade, is that I owe you a curve here. I left it out, and that's the random. And the random actually is slightly below here. And again, there are, so essentially the quick rationalization why the random is a little bit below is, so random is not a subset of this one, random is a subset of all the single possible variants. Okay? That's the way we define random. I define here random when I say the random is slightly below. I don't show you the curve. And the rationalization of that is simply the difference between what we see and what random is some cannot walk into the room to be sequenced. They die out, or they will never be born. And ultimately, the very bad variants we don't have in there, and that makes random differ from what we observe. But other than that, what we observe is pretty close to random. That's what we argue. Again, there are still significant differences, and that's part of that publication. But I owe you the random slightly below, so you would say it's below. Everybody fine with that? Hey, we have. Okay, I believe I'm going to change my view on this group. I don't have a lot of interaction, but every single time you're right, somehow right on at least with my expectation. In this particular case, you are even right on with the truth. This is where the Denisovans fall. So, slightly below. And in fact, the random would be pretty close. I haven't looked at it, but again, there are different random models. Now, when you write a paper, when you write a thesis, when you present a talk, first you explain what this curve means. So this is by now I've amply explained this curve. And then you make statements, make observations. And again, I said that light blue is below purple. What's the next thing that you observe in a publication and talk? Certainly in a publication, sometimes in a talk you don't have time for that. But mostly actually in a talk you should still say, what else do you remark on that light blue curve?
It's jumping up and down.
Why? Anybody else? Are you somebody who hasn't made an observation? What the ragged curve, what does it suggest to you? There's nothing. You should jump up. When you see ragged curves, always cry for error bars. That means that there's something, when the curve is not smooth, either there's an important biological reason for that, and then you really want to find out about that, or it has to do with just too high errors. What typically is the reason for too high errors? Or what could be the simplest thing that you could suspect? It's not the inaccuracy of the data set. It's mistakes in the data set, that's true. But that's not what I mean here. Typically it's related to numbers, right? That is sort of very often the case that we don't have enough observations. And that exactly is the case here. We have a human population of 400,000 variants. We have one Denisovan. One. Actually, why don't we have more? Why don't we sequence more Denisovans?
Maybe they didn't find more.
Yeah, all the genetic material of a Denisovan has been sequenced, eaten up by the lab in Leipzig. So they did 3D prints of the little piece they had. I can't remember, they have so many stories that I confuse the stories now. I believe it's the tip of a finger. So you have a probe that is exactly, it looks to everybody the same, but the biological material of all the Denisovans ever found is gone. It's all traced in the sequence. And so they first did the highest accurate genome sequence of any individual ever at the time they did it, and were very happy about it. And after they were happy about it, they began to, well, now we want another one, right? They wanted about diversity, but okay, they're looking. This is all we have, okay? And it's clear there's mistake in the data, or the data is too spotty, not mistake. Mistake is the wrong word. We need more. We cannot get another. Oh. I was just about to open the window. What would people say about this?
Go for it, that's a good idea.
that makes all the difference when you say that. Okay, so I suspect that blue is below purple. I cannot get another Denisovan. How could I see whether I discovered some theme rather than just, a small data set? What other experiment could I do? Not experiment, what other data set could I as a computational biologist turn to analyze and look at in the same context? Any idea? Any suggestion?
The Neanderthalian?
Yeah, so the problem is we have even fewer. And there, maybe we can use the rest of today to discuss a little bit about the issues, why this is the case.
What about Homo erectus?
Yes, let's talk about that later. So nothing that has homo in the name will work.
Chimpanzee?
Ah, great. Gorilla, pan, so chimpanzee here, gorilla. Chimpanzee is a little bit closer to us than gorilla. And I'm going to show you gorilla. The chimpanzee data I have too, and it's a little bit higher than the dark blue gorilla. So we clearly have a story. In fact, here we go to the next level. What I don't show again, this slide I don't show is flies further down. Mouse, gorilla. So we clearly have some proportional change according to evolutionary distance. The further away, the more neutral. Okay, that's the observation. So this suggests that although that is a small data set, that the data set essentially captures a real direction. And now we have a couple of issues here. The first issue, why? The second issue, I have to sort of correct some possible misunderstanding here. When I look at the purple, I compare every single protein, 20,000 proteins between us. I look at the variance between these 20,000 proteins. When I look at these here, I have a much, much smaller data set. Because now I look at only proteins that I absolutely know are related. And I need a high threshold. So the comparison to gorilla already has something that is, I can't remember what the numbers are, but is still more than half, but this is a much, much smaller set of proteins, okay? So we have more variants in each protein, but much fewer proteins. So we are talking about a subset, and that gets increasingly true the further we go down. Okay? So we're not talking quite about the same thing here. That's one reality. The other reality is that what we're talking about here is a living population. What we're talking about here, for all of the other curves, is a frozen point. There was a speciation event, they separated, there was no, for these cases here, there clearly is not any evidence of further genetic exchange that lives on in us. And that means that we essentially look at one point in time when these species, our ancestors, separated, and those are the variants we see. Okay? The things that happened afterwards, so a gain of function in a different organism, are typically things where I would say, well, I cannot recognize the similarity between the proteins, I take them out from our comparisons. So I really have a view, a biased view, in the way I do this. Okay, still, having said all of that, is still actually, to me, it's surprising. Now, in a situation like that, here I talk about the out of Africa, the out group, assume this would be the speciation event, then typically, I believe the assumption would be that there's the so-called the Cumulative neutral theory that would tend to explain that this actually is expected. So the speciation does not happen on genotypes that differ. Put differently, so it's typically not this idea that we form a new species because we have a genome that is different from everybody else. We like ourselves better because we have a more similar genome. Typically, the event happens differently. Typically, there's some sort of external event, such as the one that split bono and chimpanzee. A river, the Kenya River, suddenly happens through tectonics. Two groups of apes are split and completely develop differently. It becomes different socially, different in every respect, bono and chimpanzee over here. So that sense, this is sort of motivated. It remains surprising. In terms of machine learning, what is surprising here is, remember this green here is the development set. Now we have neutrals that are even below the development set. And that can be used for training. And again, this is something that we have, I believe, not done quite. And we still do. But let me focus on some other aspect here. The other aspect is the one that we have a high effect for many of the variants between us. And that clearly is surprising. Even for some 12% of the variants, the effects are even as strong as what I would expect from the OMIM set. So the purple and the OMIM meet at levels higher than 82. 12% of the variants are predicted at that level. Why? Now here comes another story, and that is the one from Jana Bromberg, in which she says, well, actually somewhere here we already have variants with a slightly non-neutral or slight effect. And maybe the collection of these little effects, they form our individuality. That's the paper in PNAS that Jana Bromberg published. Essentially, many weak effect variants may define individuality. And she assembled a lot of data that support that point, I believe. But let's turn again to the really strong variants. And the real strong variants that look like OMIM. Sickle cell anemia, let's look at that. Sickle cell anemia is a disease that is caused by one variant in hemoglobin. At position 6, the E is mutated to a V. That causes a bad disease. This disease mostly happens in malaria regions, and it turns out that the same disease, or the same variant that causes a disease, makes you resistant against malaria. Okay, this is an oversimplification of the story. And this is not a new story, as shown here. But the point that this story makes is that something can be good and bad. So you can have a variant that causes a disease, and the same variant can help to get a new function. This is important to keep in your mind. Variants can be bad and good for the same carrier in some sense. And now, back to those. Maybe, if we looked at the ones that are really bad, maybe some of them have a good sort of effect and some of them are really bad. What can we do in order to sort of increase the goodness of the effect? So, is there any way in which we can shield us against the bad part and thereby increase sort of the good part? Well, personalized medicine. The personalized medicine idea is an idea where I have one drug for every person. So then you could imagine that, okay, I have some of these have bad effect, but for that I get my drug. And then sort of all that remains in terms of effect in the variant pool that I have are good things. Okay? How likely is it that everybody in this room here will have a drug made and tailored? How many new drugs do we have per year? 20. All over the world we have 20 new drugs in a given year. Who contests that view?
Say it's less than that.
Oh! You guys keep amazing me. 20 less, 200, 2,000 more. So, can I have hands? So less than 20 is.
Are we talking about approved drugs on the market?
Yes.
Okay.
And what I'm going to show you is FDA, so US, Federal Drug Agency, which is not that different from. So the numbers differ slightly, but. Okay, how many would believe more than 2,000? 2,000. 200. 20. Oh, that's getting less. Ah. Does it sum up? Are we 10? Yeah, that's roughly. Good enough. Wow. Wow. You guys know too much. So this is where the data is published. And you see, this is very, very, I mean, there are years with fewer than 20. There are years with sort of just around 20. But essentially, most of you are in the right ballpark here. Now, what I want to point out, at some point here you put the age of genomics, biotechnology. And clearly, we had hoped that biotechnology would change this. If anything, you see it went down, not up. But it still is true that biotechnology is absolutely crucial. And the reason for that is just, if without biotechnology, I don't believe we would have new drugs. Okay? So yes, the number did not change very much, but it got so much more complicated that most likely without biotechnology we wouldn't have any anymore. Part of that reason is explained that it takes 14 years at this point to develop a drug. 14 years. Now imagine, and here's the second number, it takes $2 billion. Even if you were one of those, I don't know how many people have more than $2 billion to spend on their health. There are not many people. We're one of those 100 or 200 or something people in the world who could. Typically would do that only because they have a very bad disease, and maybe 14 years is too long for that. And this is an average. Not every drug, not every disease can be cancer drugs, you know, HIV. So there are tons of drugs where tons of diseases where we know there is no drug available, despite all of that. By the way, ultimately these two numbers, or these three pieces of evidence, they together totally explain why you have the merger of pharmacy. Of pharma companies, because an individual pharma company can no longer take the risk. They have to be giants in order to survive the game, and that has to do with the fact that FDA approval gets more and more complicated. It has to do with the fact that the game is getting more complicated overall to develop a new drug. And the costs for that are tremendous. Whenever you're a small startup company that has a target that gets sort of close to phase 3, to the clinical phase, then essentially you're bought up by the big ones. That explains the... But one thing that it clearly says is that there will not be a drug for everyone in the room. Okay? Maybe we get this number up a little bit. Hey, we get it up by a factor of 10. Over the last 30 years we didn't, but maybe next year we do. But still, this is not enough for what we need for the idea of one drug for each genotype. But what we can do is what I call personalized health. What diagnosis, choice, food, that we can do. So what I mean here is, for instance, you have 3 different drugs, and maybe as simple as headache. And your genotype fits better to one. So for headaches, it's easy. You just try out 3 different types of medications and fine. If you have a bad brain tumor, you get 1 shot. Typically, for some brain tumors, the survival time is so short that you get 1 trial of the 3 medications on the market. And typically that is administered because of the local physician in that local environment has gathered some experiences that maybe you look like a 55-year-old male with a big nose and a big belly, and you get that one. Okay? If you would look more like a bearded guy of 20-something, you get the other one. This is typically the way the choice is done today. Because there is no other consistent data that the physician may have integrated data on a less rational way, and that may often be very, very helpful. But that's the only way the decision can be made today. In some cases today that's already different. So in the US, some of these decisions are already done by genotype. So I have, for instance, a high blood pressure. My genotype says that certain drugs I should not take. In fact, they lead to heart arrest, and that can happen pretty rapidly. And that in itself doesn't happen very often, but the moment the genome price comes down to levels of maybe 200-300, and we're not that far away from it, that will pay off for the health industry, or at least the insurance, to pay for that sequence be done, to just avoid that 1 in 1,000 makes this mistake. This mistake costs them so much money that these 1,000 genomes will be worth it. That is something that is happening today, and more of that to come. Food, I believe, is another way, in the way we feed, the way we live. So in some sense, I clearly believe that studying the genome type and learning the effects of variants is part of increasing the goodness of this diversity that we have. So I'm going to rush through, I thought this would be faster, some other data. So this is all unpublished, hopefully be submitted on Monday. This work, first author is Jonas. And what he, for instance, looked at is OMIA. So this is man, online mammalian inheritance in man. This is animal. Okay? The number is so small not because the database is so much smaller, but the database essentially is not curated in a way in which we could just take the database and put that into our machine. Essentially, Jonas spent the entire summer reading papers and extracting the sequences from the database to come up with now a curated set of 117 variants. That is the problem. But clearly, no, not clearly, but the green curve clearly somehow we can predict the effects. And not only that, there is a tendency that it looks as if the green, again, now we have a very noisy data set here, it looks as if the green is above the red. So OMIA is above OMIM. Anything that you know that would explain this? Well, it's very simple. So people walk to the doctor because they have 6 toes. They feel this is something that is worthwhile to see the doctor for. Animal doesn't do that. So when the animal is recorded here as a disease, then those are really bad ones that simply diminish the value of the animal. So those are really worse diseases, stronger effects. That's one. That's at least what we suspect. Let's look at a humanized mouse. So a humanized mouse means that I take a variant, I have a variant in a human, and I take the entire protein of the human and put it into mouse. Okay? And I can do that in different ways. So the simplest way would simply be, I take the mouse sequence and just change the same residue. So I have like in sickle cell anemia, where I have position 6, E to V. Whatever the mouse has at that position, I turn it to a V. Now, if I did that, what do you believe, if I did that for all the OMIM variants, what do you believe the effect would be? Less or more? So I have in silico humanized mice for every OMIM in some sense. And I'm not making a prediction whether the mouse will have the same disease. That's not the point. It's a totally different immune system. Things will be different. But just the molecular effect prediction, what we see is that the mouse, in fact, has less effect. And again, we try to rationalize that. I believe there's okay, now these curves are very similar to each other. But this tends to be, again, an unexpected, for me that was unexpected. You may argue that you would see more, we see less, and again, I believe we have a reason why. Now, the positions that we observe in OMIM, so those are rare diseases where people get sick, do you believe they are random? What do you believe, how will they stand out? So, what we show here, and that is again for OMIA, okay, so, I'm sorry, this is a little bit complicated. This is the original data. This is when I take the same conservation. So I look at the OMIA variant, look at the same protein, look at how conserved that OMIA variant, how conserved the residue of the OMIA variant in the alignment is, and then I find every other residue in the same protein at the same level of conservation. Okay? Everybody clear about what I'm doing? Okay. I have an OMIA variant. I have a variant that causes a disease. I look up how conserved that variant is in an alignment between mouse or this animal and everything else. And it could be very conserved, it could be a little bit less conserved, and that level of conservation, say, is 80% conserved. Just put a number down. Then I look up every other residue in the same protein that is 80% conserved and vary that and predict the effect of that. And that's the light blue. Given the spread of these curves, or the low counts here, those curves are very, very similar. Actually, for OMIM, is that OMIM? Yes, for OMIM, the two curves are now here. So you no longer see a difference. When I just look at random differences, I do see a difference. And this does not allow me to select the variant, but I clearly see that there's a lot of conservation in this signal here. This does not help me to predict. That's a different story. But these kinds of analyses are part of the exercises. Conclusion. The most important point is we differ a lot, and these differences matter. They make out our individuality, but they also make us sensitive to problems. How can we have so many things that are potentially dangerous? Well, because we don't survive as an individuum. Evolution cares for the species. So if I get sickle cell anemia and I die from it, fine. But if my offspring live in malaria and they survive in malaria, then what is a disadvantage to me may be an advantage to the species. The sickle cell anemia is sort of one little bit pushed example, but ultimately what remains true is that evolution plays with a lot of variants that do not have a benefit directly for the individuum that carries them. But they may turn out to be the winner. They may be rare. If they are rare and they are the winner, they will become common. For the ones who have them, they're not good. And in some sense, what I'm arguing is that understanding variants, effects, understanding biology better will help us to curtail the negative effects, and in that sense, it allows us to somehow control the bad effects, and through this somehow we will be able to combine the best of both worlds, to benefit from the diversity of people as a species, and to benefit from my own genome as an individual so that I live happier and healthier. And I believe part of that really will come from studying molecular biology better, and it will come from these initiatives such as Genomics England, such as precision medicine in the US, such as digital medicine that is beginning in many places now, and as well here in Munich and Bavaria. Most of this here is, by the way, a TEDx talk. Making a TEDx talk sort of turned my life upside down. And I believe with that, I thank you for your attention. I will still talk about the questions about Neanderthal. Are there any questions that require slides? Then I will try again to switch this off.